Hello? All right, gang, time to get started. We're going to finish oxidative phosphorylation today. Then we will get into the hypoxic inducible factor and how the hypoxic inducible factor is related by oxygen and how it regulates other genes necessary for cells to live under anoxic conditions. That's the subject for today. Let's have a word of prayer and then we'll dive right in. Father in heaven, thank you once again for a great Thanksgiving, for a great weekend, for a great chance to not have a fixed schedule, to do things on our own time. Thank you for the chance to get together either by Zoom or in person with those we love. Thank you for our families. Help us to never, ever, ever take them for granted. Thank you for the chance to get an education. Thank you that it's the home stretch of this semester. Help us finish strongly, Lord. Help us stay the course. In Jesus' name, amen. And last time we looked at this malate aspartate shuttle. This allows you to take reduced molecules, put them into the mitochondria, oxidize them, send them back out, load back up, come back in. This is necessary because NADH or NADPH cannot cross the inner mitochondrial membrane. Neither can oxaloacetate. So you have to go around it. That's why you have shuttles. And this is the one shuttle that, at least in animal cells, is definitely used most. It is definitely used a lot in fungal cells too. Plant do use it. Don't think you could say they use it as much as animal cells, but they do use it. So this particular shuttle we went through last time, hugely important for getting a reductant that is made via glycolysis out in the cytosol into the mix so that it can be used for the electron transport chain to make ATP. That's the malate aspartate shuttle. Plants, as we before, have an externally oriented NADH dehydrogenase. It will take electrons and reducing equivalents from NADH, oxidize NADH to NAD, transfer those electrons and hydrogen ions straight to ubiquinone. And now ubiquinone can participate in the transport chain, taking its electrons, sending it straight to complex 3, and then complex 4, and so on and so forth. Muscles use a glycerol 3-phosphate shuttle. The reason for this is that resting muscle will tend to use fatty acids or fats in general when they're actively respiring. They will tend to burn glucose anaerobically. That's at least the case for a fast twitch muscle. So because the resting muscle will tend to use fat, that fat will be hydrolyzed. You'll therefore have free fatty acids, glycerol. Glycerol is converted to glycerol 3-phosphate by triose kinase. Glycerol 3-phosphate is then oxidized to dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase. That produces an NADH. That NADH is then sent directly to the electron transport chain via this little shuttle that you have in muscle cells, which is shown here. So you give that reducing equivalent to the flavine. The flavine will then give its electrons to the ubiquinone, which takes the electrons to complex 3. That oxidation converts glycerol 3-phosphate into dihydroxyacetone phosphate, which can be converted to glycerol 3-phosphate again if you want this to happen in a cycle. But if you are oxidizing fat because you're resting muscle, because you want to spare the glucose for the brain, this is how you will do it. So that reducing equivalent goes straight to the mitochondria. Okay, now the depletion of ATP in cells can result in cell death. So because the ATP synthase can also work in reverse, it can potentially take a damaged mitochondrion, it can pump protons from the mitochondrion and hydrolyze ATP. If this happens, you're going to waste enormous amounts of ATP. And if you deplete the ATP in the cell is going to give up the ghost and die. So there is a regulatory protein that is called HIF-1. HIF-1, as you can see, the white here is, and also the red, is a simple protein. It binds two of the F1 subunits of ATP synthase, and it locks them in place. So this will inhibit ATP synthase activity. It operates in a pH-dependent manner. It's most active at pHs below 6.5. If the activity of the activity of IF-1 will protect cells during periods of ischemia when molecular oxygen levels fall, leading to a reduced proton gradient. IF-1 will function as a homodimer. It will link the two ATP synthase complexes through interactions via the F1 beta and gamma subunits, locks them in place, prevents them from going backwards. So F1 prevents ATP hydrolysis during ischemia. There is a second regulatory protein that's called factor B. Factor B. And to explain how factor B works, let me use our little makeshift blackboard here. Oops. So in the membrane, so this is your inner mitochondrial membrane. If this is your matrix, you're then going to have the that will be something like the F-type, the F-naught, FO subunit. Remember, the protons are going to enter through this F sub O subunit. Then you have the stalk, and then you have the subunits. That looks like an impressionistic ATP synthase. All right, so you have a second protein that is called factor B. Let's make factor B just this big yellow blob like that. So the IF-1 interacts with the F sub 1 subunit. This is going to interact with the F sub O subunit. It's going to interact, and you know what? I drew this on the wrong side. It's going to interact on the matrix side. Sorry about that. I didn't pay attention to where I was drawing. It will block the leaking of protons across the inner mitochondrial membrane. So this is going to essentially bind here. It will block the leakage of protons across the inner mitochondrial membrane from the intermembrane space. The ATP synthase unit assembly factors also going to help protect the hydrophobic surfaces of the alpha and the gamma subunits. That's going to prevent them from aggregating together. So there are some hydrophobic surfaces. So this is going to, factor B, prevents leakage of the hydrogen ions back into the matrix. Also, you have some hydrophobic surfaces. These hydrophobic surfaces can cause this to clump. To prevent this from clumping, because remember, there is another protein here as well, and you have adapting proteins here. There are two assembly factors. They're called ATP11, ATP12. These will protect these hydrophobic surfaces, and I'm going to draw those in red. These are going to protect these surfaces from aggregating together. Under ischemic conditions, because the ATPase now can become relatively quiescent, these hydrophobic surfaces can cause the protein to start to clump and aggregate together. If it clumps and aggregates together, it may lock itself into a non-functional conformation. And now this complex is 1, non-functional, 2, very hard to get rid of. So these are meant to prevent this from happening. There's an additional protein called TMEM70 that also will associate with the ATPase and prevent it from clumping as well. So the ATPase is very carefully protected within the cell. HIF-1, factor B, ATP11, 12, and TMEM70.
So the only thing that you drew on the wrong side was that original factor B, correct?
Yeah, it's funny on the matrix side. I just drew
No,
it.
it's fine.
Didn't think about where I was drawing it. The factor B is on the matrix side. It acts on the matrix side, clearly.
Yeah.
It's only found out in the intermembrane space if there's damage to the mitochondrion. Right, so in other circumstances, you want to generate heat, and there are special tissues in your body that are used to generate heat, and they're called brown fat. Brown fat. Brown fat tends to be found in places that lose a lot of heat. Brown fat can be made from white fat if that white fat is exposed to cold. And so the thyroid gland makes thyroid hormone. Thyroid hormone activates transcription. One of the targets of thyroid hormone is this protein, UCP, the uncoupling protein. The uncoupling protein, when it's made, is inserted into the inner membrane of the mitochondrion, and this serves as a pore for the protons that are pumped out into the intermembrane space to re-enter the matrix without the benefit of ATP synthesis. However, this entry of protons generates heat. Generates, in fact, a fair amount of heat. There are also other types of organisms that need to make heat. For example, skunk cabbage that will tend to come up and pierce through the snow. It's one of the first flowers that sticks up through the snow. The skunk cabbage actually melts the snow a bit. because the mitochondria in the cells of the tip of the skunk cabbage sprout have an uncoupling protein. So those cells generate a good deal of heat that slightly melts the snow, and that's how those skunk cabbage sprouts are able to pierce through the snow. Also, brown fat in our bodies, we have it on the back of our neck, our underarms, tips of our elbows, places like that. They will synthesize the uncoupling protein, and they will synthesize heat. The amount of brown fat in a person can also determine how much heat they make. And if you know some people, they're just constantly warm, that might be because they have more brown fat than usual. All right. And then finally, we have cases of hypoxia. You can see hypoxia is going to induce transcription factor called the hypoxic induced factor 1, or HIF-1. And HIF-1 is going to induce the enzymes of glycolysis, but it's going to induce the degradation of elements of the electron transport chain. It will also tend to inhibit pyruvate dehydrogenase. And we will take a look next at how HIF-1 is regulated by oxygen. This is stuff that's not in your book. So I'm giving you a little extra stuff about the very cool molecule HIF-1. All right, HIF-1 is found in just about every multicellular creature you can think of. It's very highly conserved. If a species is air breathing, oxygen breathing, it usually has HIF-1 or something similar to HIF-1. HIF-1 is composed of two subunits. They're very creatively called alpha and beta. Alpha and beta. The beta subunit is on all the time. It's always there. So it's constitutively expressed, and it is the aryl hydrocarbon receptor nuclear translocator. And that's what that protein does if it's not teamed up with the alpha subunit to form HIF-1. The alpha subunit is the one that is post-translationally induced by hypoxia. Under conditions where there's plenty of oxygen, this subunit is inactivated, and I'll show you how in just a bit. So HIF-1 is a member of the per-arndt-Sim or PAS subfamily of the basic helix loop helix family of transcription factors. That probably means absolutely nothing to most of you. I will give you a little bitty primer on basic helix loop helix proteins. So let's say you have some DNA. And you have a gene, and you want to express that gene. So the gene, the coding region is down here. And this portion here is the promoter. And in order to express this gene, we need to get RNA polymerase to bind somehow to this DNA and start transcription. Now, RNA polymerases in eukaryotic cells don't do that easily at all. So they need help. And from whence their help comes? Their help comes in the form of proteins that bind to DNA and these proteins that bind DNA will provide protein-protein interactions with the RNA polymerase that stabilize its complexation on the promoter so that it can initiate transcription. So these transcription factors bind to specific sites, and those sites are typically sites that can work in either orientation, or they can work regardless of where you put them, so they are known as enhancers, because they enhance the capacity of RNA polymerase to bind and initiate transcription. So one of the groups of these transcription factors that activate RNA polymerase complexation on the promoter are the basic helix, loop, helix proteins. And these are just called BHLH because writing that out all the time gets to be a drag. Now, what's characteristic of these proteins? So these proteins have, first of all, a alpha helix, which I'm just going to draw like this. And this alpha helix has lots of positive charges on it. So this is a basic region. This is going to slide into the major groove of DNA. It's going to make sequence-specific associations with DNA, but then attached to this is a loop. And then you're going to get another helix. And this helix doesn't tend to bind DNA. So you have a basic helix, loop, helix. And then the rest of the protein. Now, these proteins always work as dimers. This binds the DNA. This helps form the dimers. So you get this situation where one piece oh, that's ugly. It's DNA. You see what I mean. You'll have one piece on the DNA, so the helix piece is inserted into the major groove. You then have the second helix here after the loop. The other protein then will have its helix in the other orientation. So it will have it'll be dimerized via this other helix. These helices, they form a coil. And then you have a loop, and then this one, helix, will be in this major groove over here. And then this protein goes off over here, this protein goes off over here. So there's your basic. So these will bind to two different sites on the DNA. They tend to bind usually as heterodimers. And this is a very, very large group of transcription factors. These are particularly important in the nervous system when it comes to nervous system development. But they're important in other tissues too. But the nervous system was one of the first places where these particular transcription factors were shown to be really important. All right, so that's the basic helix, loop helix group of transcription factors. Now, the HIF-1 proteins have a so-called per-aren't-sim domain, and that per-aren't-sim domain or pass domain is quite important for dimerization of the heterodimer. So the per and the sim genes were both isolated from Drosophila melanogaster. And then the aren't is, of course, the oral hydronuclear translocator, aren't. That's the central region of these proteins. Let me just show you really fast some of these HIF proteins that are found in other. Here's a diagram from a paper. So, oh, get in focus, would you? That's a little bit better. Focus, there. So here are some of these proteins. So you have your basic helix, loop, helix, loop, helix at the terminus. Then you have your pass domain here. Then these are called TADs, meaning transcriptional activation domains. So you have a N-terminal TAD, C-terminal TAD here. And these are different groups of these. You have this transcriptional activation domains at the C-terminus, and then the PAS is more towards the center. At the amino terminus, you have the basic helix, loop helix domain here. All right, I hope that helps. Now, okay, so the amino terminus is for purposes of DNA binding. The pass domain facilitates heterodimerization, and then the terminus will recruit other transcriptional co-regulator proteins. Here is a 3D structure of the beta subunit of HIF-1. And when they make a heterodimer, here is a crystal structure of the two heterodimers. Now, under non-hypoxic conditions, the alpha subunit has a very short half-life. It's rapidly degraded. Its half-life is less than 10 minutes. The reason that is that molecular oxygen is used to hydroxylate prolines 402 and 546 in the oxygen-dependent degradation domain, or the OD domain. And this hydroxylation occurs when there's ample molecular oxygen, that is, under hypoxic conditions. The prolines are hydroxylated by a prolyl 4 hydroxylase. And that is now, when it's hydroxylated, it's bound by the von Hippel-Lindau protein. It's then marked degradation by putting a 76 peptide tag called ubiquitin on it, and now it's destined for the proteasome and is degraded. Under toxic conditions, there's no oxygen for the prolyl 4 hydroxylases to do their job. Now, HIF-1 alpha levels increase, they then form a heterodimer with HIF-1 beta, and now they go and activate their target genes. And this picture illustrates the oxygen regulation of HIF-1, HIF-1 alpha at least. So with oxygen, you hydroxylate it, you bind the HL, the von Hippel-Lindau protein, it's ubiquitinated, it's degraded. Otherwise, none of that happens, and it forms a heterodimer with HIF-1b. It then translocates to the nucleus and activates the transcription of target genes. In addition to its regulation by oxygen, HIF-1 is also regulated by factor inhibiting HIF-1 or FI8. So HIF-1 alpha has two transactivation domains, a N-terminal and C-terminal activation domain, just like you saw on that picture I showed you. These will bind to transcriptional coactivators, such as the cyclic AMP response element binding protein, or CBP, and its partner in crime, P300. However, oxygen plus the factor inhibiting HIF hydroxylates an asparagine residue within the C-terminal transcription activation domain. That prevents an interaction between P300 and CBP. So even if it lasts long enough, it won't activate target genes. This is a second layer in the relation of HIF-1. And this is illustrated here in this picture. So normally, it interacts with these co-transactivators of transcription. If, however, you hydroxylate that asparagine residue, which is conserved, a lot of HIF-1s, HIF-1 alphas at least. Now, even if you make the heterodimer, you cannot make this transcriptional activation complex. Now, what kind of genes are upregulated by HIF-1? HIF-1 binds a 50 base pair cis-acting hypoxia response element, an HRE, and in combination with CBP and P300, it will increase the stability of RNA polymerase assembling on the promoter, and it activates the transcription of those genes. So the target genes are genes for angiogenesis, that is, the making of new blood vessels, glycolysis, erythropoiesis, or the synthesis of new red blood cells, cellular mobility, and growth factor signaling. All those genes are upregulated by HIF-1. And this picture here shows you the different target genes, and you have genes for inflammatory cell recruitment. You have genes for metabolic reprogramming, fibroblast differentiation, fibroblast proliferation and migration, vasculogenesis, angiogenesis. Fibrosis will illicitly activate HIF-1, and diabetic wound heals abnormal because it tends to inhibit HIF-1. So the HIF-1 heterodimer forms. It induces these target genes. So here are some of the target genes. So the vascular endothelial growth factor, or VEGF. VEGF is certainly one of the most powerful factors of angiogenesis in the body. INOS, that's intracellular nitric oxide synthase. Nitric oxide synthase. And then ATOM, this is a metalloprotease that's going to degrade supramolecular structures so that you can make a space where you can construct new blood vessels. SDF, stem cell derived factor. And then the angiogenesis protein targeting signaler that actually tells you where you're going to make new blood vessels. It'll upregulate integrins and glucose transporters and growth factors like transformation factor beta and collagen and fibronectin and all kinds of really good stuff. Notice it increases a switch to glycolysis, a switch to glycolysis. That's very important because glycolysis will make ATP and it doesn't need any oxygen. Because it doesn't need oxygen, you can make ATP and reductant glycolysis till the cows come home and you're going to be fine. Another that's not on here is erythropoietin that will increase the synthesis of red blood cells and so on and so forth. Now, HIF-1 plays a very important role in wound healing. Wound healing. That's the reasons, one of the reasons, why HIF-1 has been studied in such great detail. So wound healing comes in three phases. There's an inflammatory phase, proliferation phase, and remodeling phase. These three phases are not distinct, they overlap, and they're continuous. Inflammation recruits white blood cells. White blood cells have high metabolisms. They burn up the oxygen wherever they are. So because the oxygen is being hogged by the white blood cells, the deeper tissue will tend to undergo hypoxia. Therefore, they will induce HIF-1. That's good. They need to. So you will increase glucose transporter, increase phosphorylation kinase 1, that's a glycolytic enzyme, increase vascular endothelial growth factor for making even makeshift blood vessels to bring materials to the hypoxic tissue. And so you can see here, you get a fibrous clot. You have neutrophils that are in here. They're chewing up a lot of your oxygen. Therefore, these cells here, they are anoxygenic. Then as the clot now starts to contract, you get this situation called epithelialization. Epithelialization. The epithelial cells now start to move and migrate this way. Why are they doing that? Well, look at some of the things that were upregulated. Migration, aggregation, bactericidal activity. These cells, therefore, they take the hint. Start to divide, they start to move, they're going to move this way until they properly join. Macrophages are going to come in. They'll recruit fibroblasts. They will lay down matrices that increase the migration of these cells and that is a proliferation and remodeling phase. Fibrin is actually going to serve as a very good substratum for the migration of many of these cells. You're also going to get proliferation of new blood vessels, complements of HIF-1 induction here. That's your proliferation stage. Notice the overlap here. You'll make makeshift blood vessels. That's called granulation tissue. Fibroblasts will differentiate or wake up. They're going to lay down collagen and fibronectin and vimentin and other tissues. This epithelial layer skin is going to now to grow and form a mature scar, or if it's small enough, it won't scar at all. And so much collagen you had to lay down to knit these together. Down here, you have blood vessels and collagen that's laid down, these fibroblasts. Now you'll have cells essentially here that will come in and lay down extracellular matrix tissue. That's remodeling. Remodeling and then proper collagen deposition, and that will go all the way up to 30 days even after wound healing. Now, thing I want you to notice the central role HIF-1 plays in wound healing. Without induction of a lot of these genes down here, you don't get these phases here because of proper wound healing. All right, so if you use an animal model where you make macrophages that lack functional HIF-1 alpha, you have impaired recruitment into inflamed areas. You have metabolic dysfunction in the anoxygenic tissue that's characterized by decreased bactericidal activity, decreased ATP synthesis, so therefore ATP depletion. This demonstrates that HIF-1 is essential for macrophage survival in a hypoxic area that's been created due to injury. HIF-1 dependent, and I misspelled dependent there, sorry. HIF-1 dependent secreted growth factors are the ones shown here, platelet-derived growth factor and fibroblast growth factor, which are essential for fibroblast differentiation and mobilization. Transforming growth factor alpha and beta, which are essential for the transition from inflammation to remodeling. All of those depend upon HIF-1 for their expression. So no blood vessels are available in the hypoxic area, so HIF-1 is activated. It makes the stromal derived factor, angiopoietin, VEGF. This induces vascularization, angiogenesis, the making of new blood vessels, the remodeling of existing blood vessels, lactic acid in tissue, and cytokines made by migrating macrophages also induce new blood vessels. And you get tissue remodeling. The stromal derived factor, sometimes called stem cell derived factor, will summon endothelial progenitor cells that are made from bone marrow cells to form endothelial cells and vascularized tissues. And then TGF-beta from macrophages, also HIF-1 dependent. And the acidic environment due to the lactate will induce fibroblasts to differentiate into myoblasts. They help re-epithelialize tissue. Sometimes you get abnormal wound healing. if you get the formation of things called keloids, that's due to the formation of too much healing tissue. You get overgrowth and you get a keloid. And that's due to excessive fibrosis. It turns out that's due to excessive induction of HIF-1. If you oxygenate the tissue using hyperbaric oxygen, you can turn off HIF-1, because continued hypoxia leads to fibrosis. Also, diabetic wound healing. It turns out that hyperglycemic conditions will suppress the induction of HIF-1 alpha in hypoxic tissues. That leads to poor wound healing in diabetics. And 50% of non-traumatic lower limb amputations are the result of poor wound healing of diabetic ulcers. So experimental drugs that inhibit the proteasome or inhibit the prolyl hydrosylases. And this experimental drug here, MG132, is a very good drug, at least in the laboratory. It works in animals. In a rodent model of diabetes, it will prevent abnormal wound healing in diabetic mice. It has not been used in human trials yet. It's uncertain if it will be, but it does work very well in rodents. So this is a proof of concept that you can make small molecules that could potentially potentiate wound healing in diabetic patients, targeting HIF-1. And then HIF-1 is overexpressed in cancers. So in solid tumors, you have hypoxic regions, and HIF-1 is very highly expressed. In tumors, if you stain carcinomas for active HIF-1, depending on the type of carcinoma, 40 to 80% of them will show active HIF-1 in the nucleus. And HIF-1, it turns out, activity can often be a marker for the prognosis of that tumor. The more active HIF-1 is, the worse your prognosis is. So there are several different drugs that target HIF-1. Some of these drugs are actually being used in cancer therapies now. Others are experimental. But by targeting HIF-1, you can potentially take away a potent tool used by the tumor to grow under anoxygenic conditions. So these drugs, again, some are already being used, some are in clinical trials, some are still in animal trials. And here are the structures of some of those compounds. All right, any questions about HIF-1 before we go on to glutathione? No questions, all right? Let's take a look at healing oxidative damage using glutathione. If you go into health food stores, you'll find glutathione on the shelf, typically. Glutathione is a tripeptide. So it consists of glutamate, cysteine, glycine. Glutamate, cysteine, and glycine. You'll notice that the bond between the glutamate and the glycine, it is a peptide bond, but it's between the gamma carboxyl group, not the alpha carboxyl group, like it should be. So that's an unusual bond. This peptide is not made on a ribosome. It's made one amino acid at a time. So they're linked, first of all, by the combination of cysteine and glutamate. That's an ATP requiring reaction. This enzyme is gamma glutamyl cysteine synthetase that forms the gamma glutamyl cysteine. This is the rate limiting step in glutathione synthesis. Cysteine availability will regulate the rate of that reaction. The second step will link the glycine. This is glutathione synthetase. That will make glutathione. These reactions will occur in the cytosol, even though glutathione is largely active in the mitochondria. And as you can see, there is in the membrane of the mitochondrion 2 different transporters that can bring glutathione into the mitochondrion. DIC, OGC. So the business end of glutathione is the thiol group of the cysteine. When you use the thiol group of the cysteine, you link two glutathiones together and you make a dimeric glutathione. And this dimeric glutathione is written GSSG, and the reduced glutathione is written GSH. That saves pen and paper. Glutathione is used by several different enzymes to detoxify reactive oxygen species and peroxides, which are made from lipids. So the electron transport chain in mitochondria will take electrons, give them to molecular oxygen to make water. Sometimes those electrons get to molecular oxygen and they don't make water. So about 5% of the time, you're going to make something other than water. You're going to make a reactive oxygen species. The most common of which is superoxide radicals. Now, superoxide radicals by themselves don't do a ton of damage. They do some. However, superoxides can react with water and they can form hydroxide. Sorry, hydrogen peroxide. And then the peroxide can react with iron to form hydroxide radicals. That's called the Fenton reaction. Hydroxide radicals are very, very damaging. And then superoxide radicals can react with nitric oxide to form peroxynitrite radicals, which are truly nasty. And this is just a thought. Notice that copper and iron are immobilized during a heart attack. That might be one of the reasons why you want to get them out of there so that they can't make further reactive oxygen species that could further damage the tissue. So, superoxide radicals on the danger scale, they're, let's say you're playing Resident Evil. Superoxide radicals are a zombie. Pow, 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 and the zombie's down. The zombie can bite you, but the zombie's usually not going to kill you. They, however, can lead to the production of nemesis. And hydroxide radicals and peroxynitrite radicals, that's nemesis. Those do a lot of subcellular damage. Okay. Now, superoxide radicals are very effectively dealt with by superoxide dismutase. Superoxide dismutase, there's a mitochondrial version of this enzyme. There's a cytosolic version of this enzyme. There's a peroxisomal version of this enzyme. This catalyzes the conversion of superoxide anions into hydrogen peroxide. And then catalase can convert hydrogen peroxide to molecular oxygen and water. Now, what if catalase gets overwhelmed? In that case, because remember, the superoxide radical can interact with water on its own. And that can form hydrogen peroxide. So if it gets overwhelmed, then you can use glutathione and an enzyme, two different enzymes, either glutathione peroxidase, which is PX, or peroxyredoxin, thioredoxin complex, which also uses glutathione and will convert hydrogen peroxide to molecular oxygen and water. And these are both regenerated by NADPH, and they oxidize glutathione to the dimeric version, and that is regenerated by glutathione reductase and NADPH. And here's a picture of these things. So here's the first enzyme. We talked about here's the oxyreductase, thioredoxin, and then the thioredoxin reductase to regenerate it. Here's glutathione reductase and glutathione peroxyreductase. All right, that converts glutathione to the dimeric version. That's regenerated by the reductase. Superoxide dismutase converts it to the peroxide. Oops, back to water. There you are. So you have several different mechanisms of glutathione peroxidase and the peroxyredoxin, thioredoxin complex, and what have you. Yeah. Okay. Now, it turns out glutathione can also covalently modify proteins. This is a regulatory step. It's a short-term regulatory step, and the function of it is not terribly clear. It seems to lock proteins into a conformation that protects them, and it only occurs under conditions of high reactive oxygen species concentrations. So maybe this is a, hey, you shut down, let's give you this shield, and then we'll take this off so you can function again. Maybe that's what it's for. Still not clear. All right, so that is, we'll finish that on Wednesday, then get into beta oxidation, and that'll be the last topic we'll cover for this semester. And I will see you guys on Wednesday. I'll hang out here if you have any questions and go from there. All right. See you guys on Wednesday otherwise.
I was wondering about extra credit. So, does the flu shot and the natural science seminar, because I'm in physiology and biochemistry, does it count for 20 points? Do each of them count for 20 points for each class?
Yeah,
Okay. Because I think I have the points for the flu shot in physiology, but not in biochemistry, and then the natural science seminar for biochemistry, but not physiology.
I'll check that.
Okay, thank you.
It's cold in here.